For those of you who don't know me, I'm Eleni Markakis. I am the Assistant Director of CBHI, and this morning it is my great pleasure to introduce Andrew Pieper, who is Professor of Psychiatry and Neurosciences at Case Western Reserve University. He is the Morley Mather Chair in Neuropsychiatry at University Hospitals, Director of Neurotherapeutics at Harrington Discovery Institute. He's a lot of things. He got his MD and PhD from Johns Hopkins University School of Medicine. He did his research psychiatric residency at UT Medical Center in Dallas. And one of my favorite things about Dr. Pieper is that he is an Eagle Scout. Welcome, Dr. Pieper.
Thanks, Eleni, for that nice introduction. And thanks, Dr. May, also for the opportunity to present our lab's work to you today. I want to say that I'm not trying to be antisocial, but I'm having internet problems at home. And so I'm going to share the screen and turn off my video while I talk, and then I'll come back on for questions afterwards. Otherwise, I'm worried it's going to overload the system. Great. So today I'm going to focus on discovery in our lab of a neuroprotective molecule, which we hope will ultimately lead to generation of a new medicine for patients. And at the same time, I'm also going to present a couple of ways in which we have used this molecule as a tool compound in the lab to lead us in new scientific directions with respect to neurodegeneration. So neurodegeneration is a huge problem today. Conditions of neurodegeneration are currently afflicting about 50 million people worldwide. And as has already been mentioned today, with our rapidly aging population, this number is just going to escalate wildly. And in fact, it's expected to triple over the next 30 years. And unless we're able to develop effective neuroprotective medicines, this is going to result in massive human suffering and potentially bankrupt society worldwide. And so there are many approaches that people are taking. And several years ago in our lab, we took a non-traditional approach to try to see if we could find something that might be able to stop nerve cell death. Instead of attempting to recapitulate an isolated biological system inside of a test tube, what we did was we instead used the entire brain in order to identify new drug-like molecules that would help nerve cells live longer, irrespective of how they might accomplish that goal. And so this is what we did. We delivered thousands of structurally diverse small drug-like molecules with no known previous function into the cerebrospinal fluid of the brains of mice in order to see whether any of those molecules could promote the survival of young newborn nerve cells in the hippocampus. As many of you probably know, certainly as Eleni knows, because she worked in this area as well, we actually make 10 to 20 times more nerve cells every single day than we need in the hippocampus. And as a result, the majority of these nerve cells die. And our goal was simply to exploit this in vivo system of cell death to see if we could find a way to make them survive instead. And we used a very carefully selected collection of compounds that were all designed to be structurally diverse and endowed with reasonably favorable perceived chemical properties, such that if we found something that worked, it would be reasonably well positioned for drug development. And after a very long process of a couple of years of analyzing thousands of mice, we discovered that this compound here approximately doubled the survival rate of those nerve cells. And we named it P7C3 because it was compound number 3 from pool number 7. It's a very exciting way to name things. And this is an example here of the greater number of neurons that survive in an animal that's been treated with P7C3 compared to one that's received just the vehicle alone. And we got very lucky with this compound. We found a couple of other ones, a few other ones actually, that worked when they were infused directly into the brain like P7C3 did. But what was unique about P7C3 is that it actually was already able to readily cross the blood-brain barrier with no problems at all. And so that is the one that we initially have focused most of our attention on. And importantly, not only did we find that it worked in rats and mice, but it also worked in monkeys as well. And so these are brain sections from rhesus monkeys who were orally fed one of the most potent analogs of P7C3, known as P7C3A20, every day for 9 months. And the graph shows, on the left, shows the increased number of surviving neurons in the hippocampus. And on the right are immunohistochemistry examples of what we saw under the microscope with there being very few surviving neurons in the monkeys that were fed vehicle, and many more in the ones that consumed P7C3A20. And so over the past several years, we've pursued three different main directions. One, we've tried to improve the properties of the P7C3 series for drug development. Two, we've tried to figure out how it is that it works, because remember, it was discovered in a target agnostic way. And then three, we've tried to see which models of neurodegeneration it might be effective in. And it turns out that the hardest thing was to figure out how it works, which I naively thought at the time wouldn't be nearly as hard as it was. But what we now know is that P7C3 acts directly to preserve the cell's ability to synthesize the essential cofactor and energy metabolite known as nicotinamide adenine dinucleotide, abbreviated as NAD. And so in the process of repairing themselves in the face of injury or disease, neurons utilize vast amounts of NAD. This is especially important in maintenance of the axonal projections of neurons in the brain. Those axonal projections can be extraordinarily long, as well as in the spinal cord. And so they're very energy dependent. They're very dependent on prevailing levels of NAD. When the energy demand becomes too great, the nerve cells start to degenerate. And that typically begins with axon degeneration and then ultimately proceeds to death of the nerve cell itself. And the way P7C3 works is to help cells synthesize more NAD at times when it's critically needed. And it does this through binding and activating an enzyme known as NAMT, nicotinamide adenine monophosphoryl transferase. And to date, both we and others around the world with whom we've shared P7C3 have applied these compounds to various models of neurodegeneration, many of which are shown right here on this slide. And we've learned that P7C3 not only protects young newborn neurons in the hippocampus, but also protects mature nerve cells elsewhere in the brain. For example, in the spinal cord, we saw that treatment with P7C3 protects an animal model of ALS. In the substantia nigra and the striatum, we saw that P7C3 treatment protects from a few different animal models of Parkinson's disease. And recently, we've shown that P7C3 is also protective in traumatic brain injury and in models of Alzheimer's disease as well. And I think it's particularly exciting that in the model of Alzheimer's disease, we see protection of axons, protection from nerve cell death, but we don't interfere with the accumulation of amyloid plaque. And I think that's an important thing to consider because we're going to need to be, for Alzheimer's, we need to be looking outside of the amyloid hypothesis in order to develop potential new treatments. So at this juncture, we're working with a drug development team at the Harrington Discovery Institute here in Cleveland to make optimized P7C3 compounds that we can hopefully turn into a neuroprotective medicine that might be able to go into trials for patients. And at the same time, in our basic science laboratory, we're using P7C3 compounds as a tool to help us learn new things about the biology surrounding nerve cell death. And one of the key ways that we have done this recently is through studying traumatic brain injury. So TBI is caused by anything that hits the head, and it can happen at any age in our life. In young people, it's common with contact sports. In the military, it's common with explosions or combat. And in the elderly, it's especially common with falls. The worldwide prevalence of TBI is just enormous. For example, in the US alone, it affects more than 3.5 million people a year. And I also want to note for everybody that TBI is the biggest environmental risk factor for Alzheimer's disease. In fact, it's the third leading risk factor for Alzheimer's disease, with the first two being genetics and aging. So experiencing a TBI at any time in your life increases your risk of developing Alzheimer's disease by about 50% is what the statistics are showing right now. But no one knows why. And as with other neurodegenerative conditions, there are no neuroprotective medicines for TBI. In people, TBI can be caused, as I mentioned, in multiple different ways, and it's often a combination of more than just one injury. And so what we have tried to do in our lab is model the fact that TBI is typically multifactorial. And so 3 of the principal forms of TBI are shown here. That you can have a direct concussive injury from an impact. You can have acceleration, deceleration injury of the head. And then you can also have shockwave exposure from an explosion. And our apparatus combines all 3 of these forms of injury by rupturing a myelin membrane in a pressurized chamber that delivers an instant high-speed jet of air to the mouse's head, which causes a global concussive injury a bit of acceleration, deceleration injury, and then a component of blast wave exposure. So some years ago, we reported that daily administration of P7C3 compounds beginning 24 hours after injury protected mice from developing axon degeneration, memory deficits, and motor problems a few weeks later. And we were excited by this finding because we reasoned that most people who experience a TBI would likely be able to access medical care within 24 hours of their injury. But a huge patient population that's still missing is the millions of people worldwide who have suffered a TBI in the past, often decades ago, and are now experiencing chronically progressive neurodegeneration as a result. So dogma in the field has long held that there's nothing that you can do to slow neurodegeneration once you're outside of what people believe would be an acute window of treatment opportunity after injury. And recently in the lab, we used the P7C3 compound to test whether this was really the case. And this next slide shows how we did those experiments. We gave mice the same TBI when they were just 2 months old. And then we put them back into their cages and let them live there normally for a year with no intervention whatsoever, just food and water and social interaction. Since mice typically live only 2 to 3 years, we figured that's essentially the equivalent of decades in a person. And so 1 year after the injury, we then randomized the mice to receive either daily P7C3 or vehicle control every day for just 1 month. And at the end of that month, when the mice were 15 months old, we tested their memory in the Morris water maze. And remarkably, the mice treated with P7C3 for just that 1 month period of time showed fully restored memory, which was identical to mice that had never been injured in the first place. We then stopped treating the mice, put them back in their cages for another 4 months, and retested them again in the same task in order to see whether this was a lasting effect or if it would go away when treatment was stopped. When we tested them again, we moved the cues around so that it was a new experience for the mice. And we were very excited to see that the 19-month-old TBI mice that had previously received P7C3 were continuing to show identical memory to the uninjured animals. And so in order to investigate this phenomenon further, we then examined the brains of these mice under the microscope, and we discovered that P7C3 had actually stopped all of the chronic axonal neurodegeneration in the injured animals, which is shown here in the middle panels with the black degenerating axons in the TBI group. And so we were surprised by this essentially permanent arrest of the axonal degeneration. And so to get further insight into what might be going on, we turned to electron microscopy. And here you can see that P7C3 treatment allowed brain endothelial cells to repair themselves, which then we showed allows reinstatement of the blood-brain barrier. So the red arrows here are pointing to breaks in the endothelial cells that were caused by the TBI that were present over a year after the injury. And as you can see, the treated animals to the right don't have those same breaks. And what I hadn't realized before, but what we learned in the process, is that these these brain microvascular endothelial cells are actually as long-lived as neurons in some cases. And so now I understand why P7C3 might be helping, is because endothelial cells are also, like neurons, they're also critically dependent on NAD, and they have to continually repair themselves for years on end. And so what we think is happening is that P7C3 allowed the endothelial cells to repair themselves and reinstate the blood-brain barrier. We then, in the lab, studied cultured human brain microvascular endothelial cells, and we saw that we can directly protect with P7C3 these human brain cells as well from various toxic insults. And so we're hopeful that P7C3 might be able to help protect the blood-brain barrier in people as well. So to synopsize this part, we use P7C3 as a tool compound to identify deterioration of the blood-brain barrier as one possible contributing factor to chronic lifelong neurodegeneration after TBI. And to our knowledge, this is the first demonstration that it's possible to pharmacologically more or less permanently reverse chronic neurodegeneration at this very distal time point after a TBI that occurred so much earlier in the past. And so now I want to shift to another study in the lab for which we recently used P7C3. In wondering how axonal degeneration after TBI might be linked to this epidemiologically established increased risk of Alzheimer's disease, we began investigating the tau protein, which has long been known to be involved in Alzheimer's. So tau, as many of you know, it normally functions in the axon to control axon stability and structure. And it's been known for a while that the tau protein in Alzheimer's disease is not only phosphorylated, which has been the most studied, but it's also acetylated, as shown here, which is simply the addition of acetyl groups to tau. And so Min Q Shin in the lab, a research scientist in the lab, wondered, hey, if this is happening in Alzheimer's disease, might this also be happening in traumatic brain injury? So it was a very simple experiment, and we put the animals in the same apparatus. And indeed, we observed that tau gets acetylated pretty rapidly right after TBI, and that this process is blocked by P7C3. We then showed the specificity of this effect, of this protective effect of P7C3 is due to the ability of P7C3 to elevate NAD and drive sirtuin-1 mediated removal of the acetyl groups from the tau protein. So then, using immunohistochemistry, we looked further into the consequences of tau acetylation, and we saw something that we thought was very interesting, which is that normally tau is restricted to the axon by means of this tight meshwork of proteins at the junction of the cell body and the axons, which is called the axon initial segment. However, once tau becomes acetylated, it breaks down the axon initial segment, which is shown here by a loss of staining in the left panel for ankyrin G and beta-4 spectrum. And when the axon initial segment breaks down, we then observed that tau redistributed from its normal axonal distribution into these round structures. I don't know if you can see probably can't see my cursor, but into these round structures, which indicates that it's gone into the somatodendritic compartment of the cell where it's not supposed to be. We then showed in other models of TBI, not just our model, but additional models that this precipitates spreading of pathological distribution of tau throughout the brain. And again, we showed that this entire process can be blocked by treatment with P7C3 simply because of its ability to elevate NAD and stimulate sirtuin-1, which we established through using specific inhibitors of those two enzymes. So to summarize, because I know we're running short on time, to summarize a very long story, we have now uncovered a new pathway in the brain after traumatic brain injury that leads to neurodegeneration, which is diagrammed here. Briefly, we have learned that the process begins after TBI with global nitrosative stress that results in S-nitrosylation of the canonical housekeeping gene GAPDH, glyceraldehyde phosphate dehydrogenase. When GAPDH is S-nitrosylated, it then does two things. It which coordinately activate the P300 CBP acetyltransferase shown here on the left, which adds acetyl groups to tau. And it also inhibits the sirtuin-1 enzyme shown on the right, which removes acetyl groups from tau. And so this results in a net increase in accumulation of acetylated tau. That leads to breakdown of the axon initial segment and tau mislocalization out of the axon and into the cell body, which causes neurodegeneration. And remarkably, we found that acetylated tau can also be detected in the blood, and that the amount in the blood of mice correlates very tightly with the amount of acetylated tau that's in the brain after injury. And so this made us wonder, wow, could this possibly be a biomarker that could be useful in people? And so we teamed up with collaborators that we know from Houston Methodist Hospital, and they had blood samples from patients who had visited the emergency room within 24 hours of having had a TBI, and we saw that in humans as well, the level of acetylated tau significantly increases in the blood after traumatic brain injury, even while total levels of tau remain the same. And so I want to close with just one more piece of data. If you look again over at this diagram here on the left, there are 3 points in purple where we used a therapeutic agent or a genetic modification to interrupt the neurodegenerative process after TBI. First, we blocked the S-nitrosylation of GAPDH after TBI with a compound called omegapil. And so that blocks the entire cascade. I also want to point out that exposure of cultured neurons to amyloid beta peptide has been shown to cause the same S-nitrosylation of GAPDH, which is providing another possible point of intersection between TBI and AD. We also achieved this same neuroprotective effect by elevating NAD through either P7C3 treatment or studying mice, WLDS mice, which have a genetic mutation that preserves endogenously higher levels of NAD. And we saw that was protected by elevating activity of the sirtuin enzyme. And then lastly, here on the left side, we also protected animals by treating them with either salsylate or diflunisal. And these are FDA-approved non-steroidal anti-inflammatory NSAID medications. But what's unique about salsylate and diflunisal, as opposed to all other NSAIDs like aspirin or ibuprofen or naproxen, is that salsylate and diflunisal also have this ability to inhibit the P300 CBP acetyltransferase. And we showed at concentrations that were of those compounds that were not sufficient to cause an anti-inflammatory effect, but were sufficient to inhibit the P300 CBP acetyltransferase, that we were able to block neurodegeneration after TBI. And so we wondered whether these agents might be more broadly neuroprotective as well, and it just hadn't been realized. And so we asked the simple question of whether their usage was epidemiologically associated with a lower incidence of Alzheimer's disease compared to aspirin, which is an NSAID inhibitor that does not inhibit the P300 CBP acetyltransferase. And to do this, we teamed up with Dr. Feisheng Zheng at the Cleveland Clinic and queried over 7.5 million patient prescribing records and diagnoses, and we discovered that both salsylate and diflunisal are indeed associated with a significantly decreased incidence of developing Alzheimer's disease. And notably, as you can see here, the protective effect is much more pronounced in diflunisal than it is in salsylate, which is consistent with the fact that diflunisal is also a much more potent inhibitor of the P300 CBP acetyltransferase than salsylate is. And so, in conclusion, we implemented a target agnostic in vivo screen of mice that identified P7C3 as an NAD enhancing small molecule with neuroprotective efficacy across multiple animal models of neurodegeneration. We, of course, hope that we'll be able to progress this into the formation of a new medicine for patients. And we're also using it in the lab to learn new things about neurodegeneration. Thus far, it's using P7C3 has helped us identify endothelial cells of the blood-brain barrier as a therapeutic target for possibly reversing chronic neurodegeneration and cognitive impairment in long after injury. And more recently, P7C3 aided in our discovery of this non-canonical nitrositive stress cascade in the brain after TBI that induces tau acetylation, leading to neurodegeneration. And with that, I see I'm right up against the hour. And so I just want to thank and acknowledge the many people in our lab and the funding sources who contributed to this work. And I'd be happy to take any questions.
That's great. Thank you so much for that. I do have a question, and that is, so your molecule is going to be predominantly effective or predominantly found in areas with endogenous, adult neurogenesis. Do you have any idea how much gets where when you're giving it orally? So do you have, is it blanket all over the brain, gets the same amount? And what do you know about the pharmacokinetics of it?
Yeah, we know actually that it gets distributed into every organ in the body, and it doesn't become inordinately concentrated in any particular place. Other people have actually shown that it's able to protect the liver in different forms of oxidative stress, and its protective effect in the brain is not only exclusively due to neurogenesis, although we think that is certainly helping because increasing neurogenesis is going to make the hippocampus healthier. But importantly, we also are able to stop neurodegeneration of adult neurons as well. For example, the processes from the substantia nigra that go out to the striatum that degrade in Parkinson's disease, we showed that even when we administer a toxin into the striatum, which would normally cause degradation of those, that coincident treatment with P7C3 blocks that axon degeneration.
That's cool. All right. Anyone else?
Okay.
I feel bad. I feel like I'm, taking all the questions up. But I'm just wondering, as a last idea, do you have any sense of whether this might be effective in spinal cord as well? I know you probably haven't looked. There's so much going on.
We have shown that it protects the spinal cord motor neurons in the G93 mutant mouse model of ALS. And others have shown, I'm not sure if they published it yet or not, but others with whom we shared the compound have seen that it's protective in blunt spinal cord injury. So, and I think that makes sense because they have some of the longest, most energetically demanding axons. So they're highly dependent on NAD.
That's fantastic. Thank you very much. Thank you for being with us. Thanks for talking to us about your work.
Okay, thanks for the opportunity.